All right, we should be recording now. Did everybody get the message that we're recording? Okay, awesome. I got a confirmation. So hello all, welcome to the ninth Anatomy tutoring session. My name is Dr. Haggerty. I am one of the Anatomy coordinators, co-coordinators, and I just wanted to start off by saying thank you all for joining us today. And if you, again, do not want to be recorded as you're coming in, still admitting people, if you do not want to be recorded, make sure that you turn your videos off and you also mute yourself. I have a couple of announcements coming up for Anatomy. We do have our last day of registration. It's going to be Monday, the 12th, so tomorrow. So if you have not registered yet, make sure that you do register by tomorrow to make it into the 2024 competition. And then the other announcement is that you will be having an overall local competition or review for the local competition next Sunday on February 18th, starting at 6 p.m. And that is going to be run by yours truly. So myself, Dr. Peterson, and Dr. Piscura. I think if we don't have any other announcements at this time or any other comments from our co-hosts, I would say that we can go ahead and get started. Dr. Frank, do you want to introduce our first presenter?
I sure do. Give me 1 second here. Okay, well, our first presenter is Dr. Tony Gagne. Tony grew up on the beach of Cape Canaveral on Florida's East Coast. For his bachelor's degree, he studied exercise science at Florida Atlantic University. He then completed a master's in sport health science and a doctorate in chiropractic at Life University in Atlanta, Georgia. He is currently in his second year of medical school at VCOM in Auburn, Alabama. He aspires to become a family medicine physician with a specialization in sports medicine. You can usually find Tony playing sports, being active outdoors, and looking for the next big adrenaline rush, like skydiving or scuba diving. So with that, I give the platform to Dr. Gagne.
Thank you kindly. Can I get a thumbs up if you can hear me? Good, cool. So let's share. And we seeing that all right as well? Cool, I got some thumbs up. Let's go. All right. So we're going to dive into the immune system and the endocrine system. And this was awesome. I remember the first time I learned about the immune system with the lymphatics and whatnot. I remember thinking, how has nobody told me this before? Because it was like this whole hidden highway of circulatory stuff that nobody really talks about. And I was like, this is the coolest thing ever. Like, this is like a hidden circulatory system. It's awesome. So I had a lot of fun putting this PowerPoint together. So just right off the bat, the lecture objectives, we're going to be talking about two systems, the immune system and the endocrine system. Starting off with the immune, we're going to go through and describe the organization and the function of the lymphatic structures. And we will go through that and then go through and identify and locate the major groups of all of those lymphatic structures, including cervical, pectoral, axillary, inguinal lymph nodes, and whatnot. Along with, and that'll be for the immune system. Then we're going to move on to the endocrine system. We're going to go over a brief, very surface level, just go through what are the organs of the endocrine system and what might they do. And so we're just going to list the major endocrine and exocrine glands and organs and describe their locations and general functions. Okay. So starting off right at the top, we are talking about the lymphatic system. And this, again, when I learned it for the first time, it was super cool because it's kind of like the circulatory system with arteries and veins, but it helps it, but it is different and it's unique. So starting from the outside in, if you're a bug or a germ or a piece of dirt and you're going into the body, you're going to go through the lymphatic system. And the way that works is you're going to start with a lymphatic capillary. And then the capillary is going to be different from the vessel, and we'll go over that. But the capillary is going to lead to vessels. We're going deeper into the body. As the vessels go through nodes, We'll talk about what the nodes do. The nodes and the vessels are going to eventually lead to the ducts, and then the ducts bring us back home. And the ducts are going to be very central to the body. So we're starting outside with the capillaries, going through the vessels and the nodes into the ducts where we're central. And that's the overview of this circulatory system. And to start, and when we're talking about the difference between the capillaries, lymphatic capillaries, lymphatic vessels, one of the most important things that you're going to want to look at for determining whether it's a capillary vessel is it single cell and open-ended? And the reason for that is the lymphatic system, when we're starting with the capillaries, is different than the circulatory system with arteries and vessels, because this system, which I want you to focus on right here, is open-ended. The circulatory system is a closed loop system where the heart and the lungs and the arteries and the veins, they all are connected and they go around in this loop and they do their thing, where the lymphatic system, where this starts off, is open-ended. It is in the periphery, it's in the tissues, and it is open to whatever's in the tissues at that time and that location. And then we are going to close it at the end where we get central to the ducts. So lymphatic capillaries, open-ended, and they're single cell thick, and that's because we're trying to get the fluid in. And that single cell really is the key to determining whether it's a capillary or not. The function of this right here, the function of the capillaries we are just beginning the process of returning the interstitial fluid to circulation. And I have a little note down here, I added this line: the interstitial fluid and the lymphatic fluid are the same thing except for their location. The interstitial fluid is between the body, interstitial is between cells, it's in the tissue. And then if I can draw your attention to this picture here on the right side of the screen, the blue is the interstitial fluid. Here you can see it, and you can see the same color when it gets inside of the lymphatic capillary, it becomes lymph or lymphatic fluid. And lymphatic fluid, those are the same things. But what you need to pay attention is as soon as we cross this border here and we make it into a lymph capillary, this is now lymph fluid. And that's really the main difference in nomenclature there. And that's going to be at the beginning with the capillaries. Let's see. So as we go from the capillaries to the vessels, things change. The vessel looks different from the capillary. And the way you know that is now, instead of a single layer, we have three tunic layers, and that's more akin to the veins and the the vessels of the circulatory system with the three different layers. And so that's going to be one of your biggest clues, as you'll learn in the histology, between a vessel and a capillary, is we have three layers. And another thing that's going to help you is we have valves here. So the vessels are going to have valves that prevent the backflow. One of the coolest things I thought about the lymphatic system, one of the things that I thought was really cool as I was learning it for the first time, is there isn't a pump. It's an open-ended system, and there's no pump like the heart. The heart's not connected to this system. So what's moving the fluid? Like how is this working if it's open-ended and it's not connected to a pump? Well, the pump that's going to help move that fluid, because we're in the body and the interstitial and we're out in the periphery, are actually the muscles in your body. And so the muscles of the legs and the arms and everywhere are going to help squeeze and literally put pressure on the lymphatic vessels. And because there's these valves, these one-way valves that prevent the backflow, all of this lymph is going to travel in one direction, because it's not going backwards because of the valves, and the skeletal muscles are pumping, putting pressure and moving all of that lymph in one direction. And so I was like learning this, and I was like, man, that's got to be the coolest thing in the world, is that even though it's not connected to a pump, there was a mechanism that takes care of it. And so with the lymphatic vessels, the biggest things you need to know: the three tunic layers and the valves there. And the vessels really are for transport. So the capillaries, their big idea was get the interstitial fluid into the lymphatic system so that we can get ready to transport it. The vessels, their big idea is let's get this lymph back to circulation. Let's get this flowing. The lymphatic nodes here, sorry about the colors here. The lymphatic nodes are these clusters of, it is a node here that are scattered throughout the vessels. And as you go through vessels, there will be more and more nodes. But the function of the nodes is kind of like a pit stop. Like if you're like a trucker on the highway and you got to go to the weigh station or you got to get gas, these lymphatic nodes are like a pit stop where the lymph that's coming from the body, before we get too far into the body, we want to make sure everything's okay first. And there's not any bad guys or bad things in the lymphatic fluid. And so that's what the nodes are here. What I want you to pay attention to in the nodes is the function is the filters. And what exactly does that mean, or what actually does the filtering are the macrophages and the B cells and the T cells. These are both immune cells that are gonna be the, like I like to say, the police officers at a pit stop or at the weigh station. They're doing the checks and they're making sure there's no bad guys or anything, germs or anything that shouldn't be in the lymph, that shouldn't get into the body, that's there. And so as we travel through the vessels, the lymphatic vessels, we're going to stop at these nodes and make sure everything's good. And if there is debris or pathogens, the macrophages, the B cells, and the T cells, these immune cells are going to take them out of the lymph. They're going to take care of it so that by the time we get back into circulation, everything should be all good. And so you can see that these nodes are usually found in clusters around portals of entry. That includes like the mouth, the ears, and the nose, the genitalia, the nipple of the breast, all of these places where pathogens are likely to enter. And we can take care of those pathogens before they get too far in the body and cause a problem. More notes on the lymphatic nodes here. The lymph fluid is circulated through numerous lymph nodes in a sequence from peripheral to central locations. We've been talking about that. And swollen lymph nodes, I'm sure you've heard of this, swollen lymph nodes are nodes that contain activated, which is undergoing incredibly rapid mitosis, immune cells, which we talked about, B and T cells, due to the presence of a nearby pathogen. So let's say that again. Swollen lymph nodes contain activated immune cells due to the presence of a nearby pathogen, right? And as we've talked about, those are the B and T cells. They're activated now because this pathogen, this bad guy, this germ has been brought into a lymph node, and now they're going rapid mitosis to make sure we take care of that. Moving on to the lymphatic ducts. Their general function is just to get all of that lymph that we've collected. Remember, we are central now. We're away from the peripheral. We're back into the central. We're deep inside the body here. And their main purpose is going to be to get that lymph fluid back into circulation, back into where the heart is pumping so that we can get this fluid moving. We don't like fluid to pool up. We want this fluid moving. And so the ducts are going to bring that back into circulation. Here's how they do it. There's two ducts that you need to be aware of, the right lymphatic duct and the thoracic duct. The thoracic duct is on the left because the right lymphatic duct is on the right. Where these enter is important. The right lymphatic duct is going to enter into the right subclavian vein, and the left, or the thoracic duct, which is the left, is on the left, will enter into the left subclavian vein. So thoracic duct, left subclavian vein, right lymphatic duct, right subclavian vein. For me, I remember trying to remember how this was organized was a little difficult at first, but what helped me was the thoracic duct, on the left is the large one. So all of the lymph that we've been talking about from the periphery all the way over on the left side is going to be taken care of, except for where the right lymphatic duct drains. And so the right lymphatic duct, I can say that a little clearer. The right lymphatic duct is going to drain what you see here, the right side of the head, the right arm, and the right chest. The right lymphatic duct is going to take care of the right side of the head, the right arm, and the right chest here. The rest of the lymphatics, from the legs, left side of the head, left arm, left side of the chest, all of that goes into the thoracic duct. So the way I remembered this, what finally made it click for me, was the left is large or the right is high and tight. So whichever one, usually I forget one of those, but then I'll remember the other. So some days I remember left is large, that leaves the right is high and tight, or vice versa. So hopefully that's helpful in remembering that. But right subclavian, right lymphatic duct, high and tight. Left is large, thoracic duct is everything else. Both legs, left arm, left chest, left head. Okay. So we talked earlier about the lymph nodes being in groups or clusters, usually around areas and areas of entry into the body. And so we're going to talk about the cervical, pectoral, axillary, and inguinal, and I have some pictures here for you as well. But what I want you to see is around the ear here, we have the cervical. We have also, these are auricle lymph nodes here, but down on the neck as well, because this is the drainage, right? The lymph is going to drain from each side of the head down into the chest where that thoracic duct and the right lymphatic duct are. So as we go down into the drainage, we have cervical lymph nodes, and the lymph nodes are being supplied by the vessels, and the vessels get their lymph from the lymph capillaries. And as we go down, cervical lymph nodes are here, and this is where all of the lymph is being checked, right? Here we have the pectoral and the axillary as well. And so I think this is important. Why is the lymph nodes clustered here in the axilla or in the pectoral muscles here? The lymph this is a good spot for the lymph nodes to be if there was a pathogen or a disease or a germ that was caught in the arms or the hands. Like if you're out gardening or if you're doing something with your hands, your hands get beat up, there could be a germ that gets into the tissue via the hands or the arms, and then the lymph nodes, there are lymph nodes all along the vessels of the arms and everything, but here at the pectoral and the axillary is a good spot for there to be a large number of lymph nodes before we get into the chest, before we get too deep in the body. So we have the neck is a good place for the head. We have the pectoral and axillary for the arms, and we have the inguinal for the legs before we get into the body cavity. So inguinal lymph nodes here, got some pictures for you. And you can see as we go up into the body, there's more as well. Uh-oh. Oh, I'm sorry. 1 second. We just want that. There we go. Okay. So going from, that's the immune system. So overview of the immune system, we're talking about capillaries, lymphatic vessels, lymphatic nodes, down to the right lymphatic duct and the thoracic duct. That's bringing us from the outside coming in. Okay. And the moving into the endocrine system. Endocrine system's all about the hormones and their effects and exactly what is the endocrine system and what makes that up. So we'll go over their locations and their functions. Here's the overview, just like we did of the immune system. The endocrine system is going to have these 7 organs that we're going to talk about. Pituitary glands up at the top. The thyroid gland is here in the neck. Thymus is just below that at the top of the chest. Adrenal glands on top of the kidneys. Pancreas is right behind the stomach here. The uterus in pregnancy becomes an endocrine gland, and the ovaries and the testes, female and male, both of those are endocrine glands as well, part of the endocrine system. Okay, so starting with number 1, right at the top, the pituitary. The location is the brain. If we're being super fancy specific, the diencephalon below the hypothalamus. What that looks like, here's the brain. Yes, you know the brain, right here. But if we're being fancy, diencephalon below the hypothalamus is this. Here's your thalamus, hypothalamus, pituitary gland right here. And the general function of the pituitary gland, when we're talking about the endocrine system, the general function of the pituitary, this is your master controller. This is really setting the tone of what needs to be pumped out, what needs to be cut off, negative feedback loops, positive feedback loops. I listed here all of the hormones that come from the anterior pituitary and the posterior. There's two different pituitaries. Well, they're not different. There's two different sides of the pituitary. I really am trying to point out here is the command center has a lot going on. It's kind of doing the overview of the endocrine system. That's the pituitary up high in the brain, doing all of the command kind of overview of this system that we're talking about. Okay, so talking about the thyroid as we move down here, the thyroid is just anterior to the trachea, about C5. It's just in the front of your neck here. And what it does is it secretes these thyroid hormones that are known as the metabolic hormones. So you might have heard people talk about high metabolism, low metabolism, things like that. What does that mean? All of that is regulated by the thyroid. There are conditions such as hyperthyroid and hypothyroid that just, if you have too much, there's more energy being used by the body because there's more metabolism, that's more thyroid. And if you have too little hypothyroid, there's less energy, less thyroid hormones. Okay. Number 3, moving on down, the thymus is at the front part of the chest behind the sternum right here. And the main function of the thymus is to develop and differentiate T lymphocytes. That's a fancy word for immune cells. It is larger and more active in infancy and early childhood. That should make sense because as we are children and infants and in early childhood, we are developing our immune system. And so the thymus, as we get older and bigger, the thymus will get smaller and atrophy into adulthood. And that's because our immune system has developed more and there's less of a need for it. So the main idea is as we move in down into the chest here, just behind the sternum, your thymus helps differentiate the T lymphocytes. It helps create your immune system. The adrenal glands sit atop of the kidneys. I've heard them called little hats for the kidneys, and they do kind of look like little hats for the kidneys. They sit right on top of them, and they function for fluid balance, blood pressure, stress response, nutrient uptake and release. That's a lot of words. This is a lot better way to remember it. If you think of kidneys, you might have heard of GFR for the kidneys. If you think of the adrenal glands that sit atop of the kidneys, GFR stands for the different layers in the adrenal cortex, which are the glomerulosa, fasciculata, and reticularis. So again, let's slow it down. Adrenal glands on top of the kidneys. Kidneys, you've heard of GFR. The GFR for the adrenal glands on top of the kidneys are glomerulosa, fasciculata, reticularis. Those are just the layers that are in the adrenal glands, and the different layers do different things that you can remember as salt, sugar, and sex. And what does that mean? The zona glomerulosa, the G, glomerulosa, produces hormones because we're talking about the endocrine system, which is all about producing hormones. Glomerulosa in the adrenal glands, G, produces a hormone, aldosterone, that regulates salt in the body. So adrenal glands, glomerulosa, salt. That's what you need to know. This F, the adrenal glands, fasciculata, does cortisol, glucocorticoids. That's sugar. Gluco, right, and glucose, and corticoids is a hormone. So sugar hormones, essentially, you can see here, regulate glucose metabolism. The fasciculata, sugar. So let's go from the top, adrenal glands, glomerulosa, GFR, glomerulosa, salt, F, fasciculata, sugar, and R is reticularis for sex hormones. And that's going to be the DHEA, which will eventually turn into testosterone or estrogen, depending on the pathway. But the easiest way to remember this, the adrenal glands sit on top of the kidneys, GFR, salt, sugar, sex. That'll be your best bet. Moving on to the pancreas. The pancreas sits a little lower than the adrenals and is behind, it's posterior and inferior to the stomach here. So behind the stomach. And this is cool. The pancreas actually does endocrine and exocrine functions. The endocrine means it's going into the blood and it's going to be hormonal of some type. Exocrine means it's going essentially out of the body. The way I like to think of exocrine is it's going in a duct, and endocrine goes into blood. So a duct is like a highway for stuff to get out of the body, if you will, and endocrine is in the blood that's still in the body. So for the pancreas, it has endocrine, which is blood hormonal, and exocrine functions, which is outside the body. Exocrine is outside via a duct. The endocrine function of the pancreas is to help regulate blood sugar. And it does that via secreting glucagon, which is the hormone, and insulin, which is another hormone that will help with blood sugar, right? So endocrine in the blood, in the body, pancreas is going to help with glucagon and insulin. Now, staying with the pancreas, the exocrine function is going to be digestion. If you see here, this is where it is in relation to the adrenals in the other parts of the endocrine system. But this picture here shows where the pancreas is in relation to the gut, the GI tube. And so what you can see here is the pancreas has a duct, which is exocrine, going into the GI tube that will help with digestion. So as food leaves the stomach and goes into the GI tube, we're going to get some help with digestion from the pancreas. And remember, digestion duct is exocrine outside, endocrine again, blood sugar, glucagon, insulin in the body, blood. Are we good on that? I think that's super cool that it does both. For the pregnant uterus, the uterus becomes a endocrine hormone or part of the endocrine system during pregnancy, because it's going to create the placenta, which will help secrete the human chorionic gonadotropin. For the uterus here, the ovaries and the testes, this table, I'm going to have it on all the slides, is going to help take care of everything you need to know for the uterus, the ovaries, and the testes. The uterus is in the pelvic cavity, so it's below the abdominal. It's in the pelvic cavity here, medial to the ovaries. That helps because when we go to the ovaries, as you'll see, the ovaries are lateral to the uterus. The uterus is medial to the ovaries, okay? The general function again, placenta, HCG, and you can read about the placenta and the HCG and what that does exactly here, but this whole thing is going to take care of you. For the ovaries, right? Again, we're in the pelvic cavity. We're talking about a female lateral to the uterus. So we have the uterus in the pelvic cavity. The ovaries are going to be lateral to that. And the ovaries are going to produce and secrete estrogen and progesterone. And exactly what that does right here, ovaries, estrogen, progesterone, and what you need to know right here. Okay. And lastly, for the male, we have the location in the scrotum here. That's different than the uterus and the ovaries. Pay attention. Yes, yes. And the general function is to produce testosterone. And here you have it, testes, testosterone, and their effect. Okay, that should take care of the endocrine system for you. Those are my references. Do we have any specific questions for me? I do see one, Michael.
I've heard the hypothalamus is the master gland as far as like, doesn't it secrete a bunch of hormones that then go to the pituitary and stimulates the pituitary to release hormones that control everything?
Yes, sir. You have heard correctly. That there is a thing called and doctors help me out, right? But there's a thing called the HPA axis, the hypothalamic pituitary axis, and both of those things are going to work together to command center the and let's see if I can get there for you. But the HPA, the hypothalamic pituitary axis, is going to work together to function as that command center. So you did hear correctly. Anything else to add to that I might have missed?
I just always say to my students, every every control center has a supra control center. So you may be the master regulator, but you also always have another master. And so the hypothalamus is the master of the pituitary gland, and it's also pituitary also influenced by the limbic system, which we talked a little bit about, Michael, when we were talking about central nervous system. And that's probably all you need to know for the local competition questions.
It's always a bigger
Yeah.
fish.
Yeah, that's like a really cool. Limbic is like emotion. That's interesting. Oh, for the adrenal cortex, not cortex, sorry, but the medulla layer, does that control like epinephrine, norepinephrine secretion?
That is correct. Yep. And you can see that down in the bottom right here. Hopefully you can see that, I think. The medulla is going to take care of epinephrine and norepinephrine. That's correct.
And I think Gabe Michael is going to talk a little bit more specifically about that and show you ways to differentiate these different layers, the cortex layers from the medulla, and talk a little bit more about that. So that's coming up very soon.
Okay, cool. And then last question I had for now was for someone who's like paralyzed, like paraplegic, like half their body, like their legs and that sort of thing. Do they have like is like edema problems? Like, is that the only thing that's kind of pushing the fluid in? Um, yeah, like how bad is the edema from or the swelling from the fluid for people who are paralyzed and don't have that skeletal muscle to push it into the lymph circulation?
That certainly can be a contributing factor. Whenever there's not muscles to help the lymphatics, there is like a thing like the respiratory pump where different pressures as you take a breath in, but that's super advanced and it's super small. It's not that big of a contributing factor. But yes, absolutely. Without motion, without the muscles moving, the lymph does take a hit in mobility. And it could contribute to edema. You're correct.
And I'm sure Dr. Frank is going to jump in here momentarily. But there are different kinds of stockings that, with the help of an assistant, a paraplegic can put those on. Some of them are electrified and they actually massage and move in an upward motion. Those kind of different contractions of the stocking to help move some of that lymph fluid back up towards the abdominal cavity. But I'm going to let Dr. Frank give you more details on that.
Well, I was jumping in because there's another question too. And I think it goes with this slide, Dr. Gagne. William is asking, is there any reason for the asymmetry of the ducts? And I'm assuming they mean the right lymphatic duct versus the thoracic duct.
If anybody has a good answer for that, please hop in because I don't know. And it's one of those things that usually you see you see the symmetry in the body, right? And this is one of those things that's not like that. I assume it has something to do embryologically, maybe that I'm forgetting. But fill me in, please. Help me out because I'm not sure off the top of my head.
Well, I know that embryologically there are two thoracic ducts. There's two ducts coming up through the thorax, and at some point, part of the left there's a communication between the two ducts, and the left lower part kind of goes away, and the other part separates from the right side, and so we get that transition from right to left at approximately the sternal angle of Louis, and that's why the thoracic duct looks like it takes a kind of a angulation from right to left at that sternal angle to go up into the confluence of the internal jugular vein and the subclavian vein, whereas the right duct ends up being on its own, again only doing the thorax right head and upper extremity, and that stays separate. So if Dr. Peterson or Dr. Haggerty wanted to add more to that, please.
I think that's enough detail for, for that question, but you did a great job, Dr. Frank. Thanks.
I appreciate it.
And we
Okay, are there
also-
any other questions before we move on? Okay. And in keeping with our time here, oh, we do have another one. Carlos, if you want to unmute.
So sorry if you already mentioned this, but you said that things go inside into the lymphatic capillaries.
Yes, in a way,
Uh,
yeah.
Do things go out of them as well?
They can, depending if the fluid is, or if the pressure becomes too much. We're talking about the capillaries here. And if we're looking at the capillaries, we have these, if you look here at this arrow here, when we're talking about a single cell layer, open-ended, it is possible for the fluid to build up here to the point where it goes the other way, because there isn't a valve that's a backflow valve here. And that's, I think, what Michael was saying earlier about edema. Edema can build up, and that's essentially what edema is. Help me out?
I'm just going to jump in, Tony, if you're
Yeah.
okay with this.
Yeah, of course.
Carlos, great question. And it was great to email you back and forth today. So glad that you're here in the tutoring session. So in those lymphatic capillaries, if you can see my hands, they're kind of like shingles on a house, so they overlap. So once fluid pushes in between the two, once it's inside, it does kind of close those, it pushes against the cells and closes those openings off. But I think what you might be trying to get at is once that lymphatic fluid moves into a lymph node, the activation of some of those cells will stimulate the production of antibodies, which have to move out of the lymph node, still being carried in lymph fluid, and ultimately return to the blood so those antibody molecules can circulate systemically. Even better than that, some of the cells that are kind of sequestered in the lymph nodes can move out of a particular lymph node, kind of ride the lymph highway back to the heart, jump in the blood highway, and then move back to where the pathogen is raging the infection. So there is a way to kind of transition from one, as Dr. Gagne said, from one highway system, which is lymph, to the second highway system in our body, which is blood. So it's a nice sort of interconnected system. So that was a great question.
Thank you. That was actually exactly what I was asking. Thank you.
And one last question, Michael. Make it
Sorry,
quick here.
yeah, I was actually going to ask for why is it a pregnant uterus? Like, I noticed it was listed pregnant uterus, or is the uterus always kind of an endocrine gland? Or is it just considered one during pregnancy? I remember in one of the slides.
Yeah, the short answer from my end is I believe it just when it becomes pregnant with the development of the placenta, then it becomes specific to the endocrine system. But if there's any more detail.
Just really quickly, because it is a little confusing, Michael. So the ovaries produce hormones that then circulate via the bloodstream to target cells in the uterus, but the uterus in and of itself doesn't produce any hormones unless it's a pregnant uterus.
Okay, thank you.
Okay. Well, Dr. Gagne, very nice job once again. Thank you for your explanation and expertise. And in keeping with time, we're going to move on then. And our next presenter is Gabriel Costa, and he's going to be talking about the histology of the lymphoid organs and integumentary system, or what we kind of call the skin. So Gabriel is in his second year at Drexel University College of Medicine at West Reading. He graduated from the University of Chicago, where he studied political philosophy. After a number of years as a carpenter in rural Vermont, he decided to pursue medicine and obtained a master's in medical science from the University of Vermont. When they are not studying or working, he and his wife, Sarah, are with their children. Currently, their 5-year-old daughter wants to be a ballerina, trash picker-upper, and their 8-year-old son wants to be a subsistence farmer guitar maker. Gabriel loves the three-dimensional nature of anatomy and is in awe of the embryological development. So I should have let you talk about the thoracic duct. My bad.
Oh, no, no. Can you hear me all right? Okay, great. All right, so we'll just pick up from there. I think more recently my son wants to, I think he wants to fly rockets. That seems to be what we're doing on weekends, is those little SDs, like model rockets a lot. So it's a nice little change of pace. So we're going to cover the histology of the integumentary system and the lymphoid organs today. And these are the integumentary system objectives. We'll cover these first, then we'll move on to the second half. So we describe the two major layers of the skin, including the epidermis and the dermis, and correlate with their histology, their location, their function. And then we'll correlate the histology of the hypodermis with its location, function, and why it's excluded from the skin. So when we think of the integumentary system, it's what we generally think of when we think of skin. It serves to protect the body by providing a barrier between the inside and the outside. And broadly speaking, the integumentary system is composed of the epidermis, which is situated at the surface or superficial, and the dermis, which is deep to it. So if we take a look here, we can see our epidermis up here along the top, and here's our dermal layer in this little cartoon. The epidermis is what provides that kind of physical barrier, while the tissue of the dermis is where the majority of the surface structures associated with the skin are situated. And the hypodermis, but despite its name, is not part of the integumentary system properly, but serves as a connection layer between the skin and the underlying fascia. So with that cartoon from the prior page as a starting point, please observe these examples of skin. So you need to be aware that the magnifications are not precisely matched. So kind of like a direct comparison cannot be made on like size measures here. But if we start and kind of go from left to right, the bright red image on the left is an example of thick skin from the sole of the foot. You can see that in this example, it doesn't contain any hair follicles or anything else. It's the thick skin. So we think of like the sole of the foot and the palms of the hands. And then these lighter staining slides on the right are both examples of thin skin, which is what's found on most of the rest of the body. And they have abundant structures, which we can kind of see in this layer, the dermal layer. And these represent two different skin tones, with the darker staining keratinocytes demonstrating variation in the melanin granule concentration between these examples. So moving on now, here's the histology and the function of the epidermis. It's like I said, it's the outer layer of the integumentary system. It's primarily composed of layered keratinocytes that progress from deep to superficial over time as the surface is worn away and replenished by constantly replicating cells beneath. And importantly, the epidermis does not contain any vasculature, right? So if we use our cartoon here as an example, we don't see any blood vessels or mixed in. It's just layer upon layer of cells that, as new ones grow underneath, they kind of push outwards. And I find it easier to think of these things going from sort of like the surface layer that you can look at your hand right now and see, and just imagine going deeper and deeper as we talk here. So, and as a comparison point, I've got this electron micrograph photo on the right, which gives us a little bit of a more detailed view and helps to tie some of the images from both the cartoons and the histology slides from the prior couple of slides on this presentation. So the superficial layer here is called the stratum corneum, and it's in contact with the outer environment. It's composed of denucleated cells. So what we can see in the cartoon, you don't see any nuclei in those cells. Same here, there are no visible nuclei in the electron micrograph. And another way of thinking about denucleated keratinocytes is that they're dead. Depending on where in the skin you are, there are anywhere between 15 and 30 layers of these cells. And these just kind of form this protective outer layer. The second layer is the stratum lucidum. And if I just jump backwards quickly, we can see the difference between thick skin and thin skin. And the defining difference between them from a histology standpoint is that thick skin does contain a stratum lucidum, while thin skin does not. So here, that gives us that example right here. We do not see a stratum lucidum particularly well visible on this, which would imply that this is thin skin in our electron micrograph. So as with the corneum, these cells in the stratum lucidum lack nuclei and are densely packed, and they contain this lipid-rich protein, which gives them a more transparent appearance. Deep to the stratum lucidum is the stratum granulosum, and it's called that because of the lamellar granules, which we can find in this layer. You can see them kind of on the electron micrograph here. They're obviously in the cartoon on the left. You've got like a pretty big view of them there. But if we take a closer look, they're approximately 3 to 5 layers deep, and they're less flattened than those found in the lucidum or the corneum, which makes sense because they still have their nuclei. And they contain large amounts of granular protein, keratohyalin, and fibrous keratin. Deep to that is the stratum spinosum, which tends to be is about 8 to 10 layers thick, and it's comprised of these keratinocytes with some dendritic cells that can engulf pathogens and damaged cells mixed in there. And finally, we reach the stratum basale or basal, and which comprises the deepest layer of the epidermis, and it sits right atop that basement membrane. And this is a single layer mostly composed of basal cells, which are the precursor cells of the keratinocytes. And they're undergoing constant division and supply the more superficial layers, pushing the layers up as a new one is formed at this level. You can think of like having bubbles forming and pushing layer upon layer upon layer of bubbles up as you create bubbles down below. I've got kids, so I think of this in terms of like kids blowing bubbles into milk right now, where you can kind of get a layer after a layer after a layer building up. So at this, amongst the basal cells here, there are 2 other types. The Merkel cells, which are the sensory receptors for touch, and the melanocytes that produce melanin, and which is what gives the skin and hair its color and protects the epidermal keratinocytes from DNA and from UV damage. So drawing from those examples a couple slides back, we can observe the thick skin on the left and the thin skin on the right. In both examples, the stratum corneum can be visualized at the top here. So right, if we, in this case, if we look at 4 here and 5 here, there's our stratum corneum. And then the stratum, and in this one, we can see actually here the stratum lucidum, and we do not see it on the right-hand side, right? So there's our defining difference there between thick and thin skin from a histology standpoint. That's what you want to keep an eye out for. The stratum granulosum, we can see in this first layer of cells. So here, as marked as 3 here, 3 here, can be compared. And under that, we find that thicker stratum spinosum. It's kind of this 2. And finally, it's a little tough to distinguish here, are the melanocytes from the basal cells if we look at that dense basal layer. So a mnemonic that we can use to try to remember this, if we go from superficial to deep, we've got corneum, lucidum, granulosum spinosum, and basale. And one of the common ones for this is come, let's get sunburned. So come, let's get sunburned, corneum lucidum, granulosum spinosum, and basale, or basal. Moving on, so now we've covered the epidermis, let's move to the dermis. And this is comprised of a papillary and a reticular layer. And critically, unlike the epidermis, it is vascularized. So there are blood vessels running through here and supplying the cells more directly. And at left here, we can see the papillary layer as it's indicated right here and visible at right here. So this is also kind of our papillary layer, with these in and out sort of sawtooth appearance or finger projections. And these are called dermal papillae, and that's where the name papillary comes from. So these little look like little fingers pushing up and in to that epidermis. And these projections into the epidermis contain fibroblasts and adipocytes, or fat cells, and many smaller blood vessels. Like the one just above the AR here, I believe that's a little capillary, and there should be another one right about there. If you guys can see those up with my pointer. And there are also some sensory receptors and lymph vessels that kind of move in this papillary layer. The reticular layer is thicker and comprised of dense, irregular connective tissue. If you guys remember, we talked about connective tissue a few sessions back. You can see the irregular pattern of this tissue in both images. And note that in this layer, it contains many blood vessels as well. So we've got a lot of vascularization in there. Reticular means net-like, and it's largely comprised of elastic fibers with a collagen lattice network that provides strength. And the elasticity of this layer that we see in this irregular pattern allows for movement of the skin in many different directions. And that gives skin, it kind of allows skin to have that flexible characteristic we're familiar with if you've ever pinched your skin or moved it around on top of your hand. And the collagen helps keep it from shearing apart. So you can move your skin around without just separating it. Finally, we have our hypodermis. So it's not part of the integumentary system, and it anchors the dermis to the underlying fascia, and that fascia is part of the underlying musculature and helps to allow sliding movement between the integumentary system and the musculature underneath it. It can be a little bit difficult to distinguish. I certainly have trouble with it seeing the hypodermis. It's more of this kind of fatty layer here with our dark line showing us this level. So if we see up here, we have our epidermis with the dermis underneath it. And then this is kind of our hypodermis layer. Deep to this would be a fascial layer with underlying musculature that didn't make it into this section. And the adipose layer or the adipose tissue found in this layer is the principal storage region of the fat in the body, and the hypodermis is considered a separate form of tissue. And so it's not. It's that kind of fatty layer. So just to recap here, we go avascular epidermis at the top, visible stratum corneum, descending down through the stratum basale. Got the papillary and reticular layers of the dermis with their vasculature and fibrous appearance, and then the adipose, and it is also vascularized hypodermis. So that covers part one. Let's dive into part two here. So our lymphoid system objectives are right here. So we're going to compare the histological organizations, location and functions of the diffuse lymphoid tissue versus lymphoid follicles versus lymphoid organs. And Tony did a great job covering kind of a lot of this material from a kind of a larger standpoint. So I'll zoom over some things here. Just a quick definition as a recap. The lymphatic system is a component of the circulatory, immune, and metabolic systems. It's composed of this lymphatic fluid, the lymphatic vessels, and the lymphatic cells. And there are three primary functions for it. It's the maintenance of the fluid balance, which Tony did a really nice job of talking us through, and the facilitation absorption of dietary fats from the GI tract, which we're not going to spend much time on. And the third is kind of this enhancement and facilitation of the immune system. And again, Tony covered that really nicely. So we'll differentiate the lymphatic system into primary and secondary organs of the immune system for our purposes tonight. And the primary organs include the bone marrow, where the immune cells are produced and B cells mature, and the thymus, where T cells mature. The secondary organs are the sites where immune cells can interact with antigens that include the spleen, the lymph nodes, the immune tissue, and the digestive tract, including the tonsils in the mouth and Peyer's patches in the intestines. And just a little trip down memory lane, we previously discussed the differentiation of the hematopoietic cells from stem cell precursor and covered the myeloid branch that goes on to form our platelets, the red blood cells, the macrophages, the basophils, neutrophils, eosinophils, all the phils. But the cells of the lymphatic system form the other branch of that hematopoietic, that blood forming via the lymphoid stem cell precursors, and that form lymphoblasts, which can then differentiate into the natural killer cells or the small lymphocytes. And small lymphocytes become T or B lymphocytes. B lymphocytes again then also go on to form plasma cells. And I just have a little chart here, which hopefully will be in the recording if you guys wanted to just have that on hand. So if we take a look here, the primary lymphoid organs, including the bone and the marrow. We talked about bone histology back in October, so I'll focus on the thymus this evening. So located between the sternum and the aorta, here we've got our thymus, which we can see is sort of overlying the heart tissue here. And the thymus is encapsulated by or is kind of in this fibrous capsule. And deep to that lies the cortex. And you can see here some examples of the trabeculae that penetrate the cortex. So if we take a look, we've got these trabeculae going in, we've got the fibrous capsule along the outer edge here. And again, here's a cartoon version, which gives you that vision of the trabeculae heading in down to blood vessels with the fibrous capsule on the top, cortex. And then the cortex contains these thymocytes, macrophages and dendritic cells, as well as some epithelial cells. And please note that the cortex stains darker than the surrounding tissue because of how densely packed these cells are. So again, if we go over and look at right here, we can see the cortex is this much darker staining pattern that just has to do with the density of the cells coming together. So in most of the stains that you'll encounter, this is the kind of pattern that you will see. Deep to the cortex lies the medulla. The medulla stains lighter because it is less dense. It contains thymocytes and dendritic cells and also some epithelial cells. And in this image, we can see that difference in staining. I find these to be somewhat helpful because somebody's gone ahead and sketched the sort of a copy back and forth. So it gives you a nice comparison to have an eye out for what to look for here. So here's our nice trabeculae coming in. There's our capsule, which we can't see very well in this image, but we can see the cortex here, this upper layer, and then the medulla is this lighter staining layer beneath. So taken again, here's a series of these kind of increasingly high power magnifications of the thymus. So if we move clockwise from the top, we can just observe that light and dark staining pattern. And the function here is the maturation of T lymphocytes. And it's a process that, as Tony discussed, it's complete by the time we hit puberty. And so then the thymus will regress. So this tissue would be from earlier. This is when it's still kind of in that maturation stage. And again, we can see those characteristic trabeculae coming in. We can see the cortex as this darker staining, the medulla underneath. And if we go up, I think I zoomed in kind of on this area right here for this image, our last one. So you can see that there's kind of this lighter staining region, but then the very densely packed cortex. Moving on to lymph nodes, they're abundant secondary lymphoid organs, and they're found throughout the body. And they serve to demonstrate kind of the fundamental features of secondary lymphoid organs. So again, as with the thymus, we have this thick connective tissue capsule at the outer edge. And then there are these follicular areas with a germinal center comprised of rapidly dividing B cells and plasma cells. And there we can see that we have this darker staining, denser germinal center with this lighter area in the very center, in the middle. And then here are a couple of others. So I think that this image, I zoomed in on this germinal center here. You can see another examples up here of this overall. And the function, just to restate here, the function of these lymph nodes is to monitor the lymph fluid for the presence of pathogens. So referring back to Tony, you talked about how these various regions of the body drain through specific lymph node areas. So these will pick up kind of warning signals or see pathogens draining from particular regions of the body. And at the core of the lymph node, there's this reticular fiber rich core, and that has a lot of macrophages in it. Lymph nodes have these separate inlets and outlets through which the lymphatic travels before it is filtered through the node. So if we look at the cartoon here, we can see those afferent vessels heading in. The fluid enters an efferent vessels where that fluid then empties out. And the follicles and lymph nodes are located within the cortex as they're labeled above here. So we've got our cortex, our follicles, and an individual follicle. Yeah, we just talked about that a little bit, that germinal center with that follicle. Lymph entering the node through the afferent vessels passes through the subscapular sinus. I think you can see it on the cartoon up here. And again, we're kind of looking in this region, and then drains into the medullary sinuses, and then it can pass on to the efferent lymphatic vessels onto the rest of the body. So the spleen is a secondary lymphoid organ, and it's located in the upper left quadrant of the abdominal cavity, and it contains, so here, if we're looking at somebody, this skeleton cartoon is facing us. Your liver's on your right, your spleen's on your left. And it contains these abundant macrophages and dendritic cells and serves as a principal site of immune system responses to blood-borne pathogens, because the spleen, at some point, most of the blood in the body passes through the spleen on a relatively regular basis. Blood enters the spleen via the splenic artery, and then divides into these arterioles. So I think we can kind of see our splenic artery coming in. Here's an arteriole as it comes out. Here's a venule. The arterioles enter white pulp, which is similar to lymphatic follicles found in lymph nodes, and the blood diffuses through the white pulp into the red pulp. And the red pulp contains dividing B cells and a surrounding layer of T cells. So if we take a closer look here, red pulp is comprised largely of these reticular fibers and interspersed macrophages. If you observe the central arteriole in this electron micrograph, so here's our central arteriole with this germinal center type surrounding the B cells here, which is white pulp. Unlike the other secondary lymphoid organs, we don't find any plasma cells in here. It's just those B cells. And if you look at the full color image on the right here, demonstrating kind of a cross section of the spleen, we can see again the white pulp here right around where there are some arterioles with the surrounding red pulp. And if we step back and just sort of think about these things more holistically, the white pulp can be understood as a site of kind of specific and targeted immunity development, while the red pulp is kind of this non-specific filter that exists outside of it. So then our last component here is lymphoid nodules and diffuse tissue. So if we look on the bottom left there, we can see an example of tonsillar tissue. And these are located at the rear of the oral cavity, visible in this photograph. So if you, so here's some palatine tonsils, which you can see if you had somebody open their mouths up wide and say, ah. And note that in this, we don't see any encapsulation. And they contain these crypts lined by stratified squamous epithelium, which allows for the intake of matter that gets ingested during eating and breathing to be then acted on by the immune system. In the center here is, if we move along the GI tract, is mucosa, so it's lymphoid tissue, or MALT. This example is a slide taken from small intestine. It contains these lymphoid aggregates known as Peyer's patches. And we can see they're roughly dome-shaped. Peyer's patches are lined by specialized M cells at the lumen of the intestine, and these M cells serve to sample the contents of what's in the intestine and transport these messages to the follicles. Different forms of mucosa-associated lymphoid tissue, MALTs, can be found in tissues of the breast and the eye, as well as in the lungs. And finally, we can encounter these diffuse lymphoid tissues. Unlike the more regularly structured secondary lymphoid organs we've talked about, this unencapsulated tissue does not have clearly demarcated boundaries. And instead, you're going to look for these patches of darkly staining cells located within other tissues. In this instance, the diffuse lymphoid tissue, this blue staining region up here, is in the wall of a bronchus of the respiratory system. And we can see that this is this respiratory epithelium on the lower part of the image here. So here's our little diffuse lymphoid tissue bundle. And that does it for tonight. I know that was a lot in relatively short order. There's some references, and please, if you have any questions, let me know.
Hey, Michael, go ahead.
Okay, I'll start with what I'm most curious about is you mentioned like those B cells are dividing and it's developing immunity. Could you talk a little bit about that process?
Let's see, we want to go back to here within, I think it's beyond what you guys need for this section, but Dr. Peterson, I don't know if you wanted to step in and kind of talk through kind of a broader look at.
Sure, if you could go back to maybe the lymph nodes, since Michael, you specifically wanted to know a little bit more about B cell activation, is that correct?
Yeah.
Right. So when and Gabe, if you've still got, your annotation tool going, if you would just show everyone the germinal center again. So these are sites in the cortex of the lymph node that are rich in quiescent, or what we just quiet, inactivated but mature B lymphocytes. And typically the process is that a pathogen is brought to the lymph node via the lymph circulatory system through a lymph vessel. And as it percolates from the cortex towards the medulla, as it's moving inward, those pathogens will trigger specific, out of the billions of B lymphocytes in that lymph node, a small handful of specific B cells that recognize that pathogen and only that pathogen. And once they become activated, then they undergo this rapid mitosis and specialization to become an activated B lymphocyte, which Gabe mentioned is called the plasma cell. And this is phenomenal. A single activated B lymphocyte or plasma cell actually produces on the order of hundreds of thousands of antibody molecules every minute. And those are getting dumped into the lymph fluid, circulated to the bloodstream, and then they'll circulate back to the area where that pathogen first entered the body. And what antibodies do, they're kind of like, I used to say to my undergrad students, they're a flag and a tag. So they flag and tag the pathogen, making it easier for macrophages to then phagocytize those pathogens. So that is way more than any of you will need to know for the local competition. But maybe having a little bit more of the detail helps you at least focus in on the cortex and the significance of those germinal centers where the B lymphocytes are localized to.
It's phenomenally cool.
Another question, Michael.
Yeah, no, that is really cool. Thank you. Next question. What are trabeculae? Because I noticed they're like kind of extending in to a lot of these organs. Like, what do they do?
So again, we have them here. If we look, here's an example of one coming in the edge here. Let's go back. Here we've got a couple harder to see in this image. And here, and they allow for, if we look, they allow for this kind of communication down to the blood vessels at the base. And that's more or less what I got for you. Again, any of the faculty are welcome to step in at this stage.
I can step in for this one. So it's actually to keep it for like simplicity. If you notice, Gabriel, if you can point out the capsule in your image,
right
especially
here.
in the bottom image on the right-hand side.
Yep, down here we've got our
Yeah, see
capsule.
the capsule and then the
trabeculae.
trabeculae. So oftentimes that connective tissue that creates a capsule will invaginate itself into the parenchyma or the functioning tissue, right? And we oftentimes call it a trabeculae. Sometimes they're called septa. Septa just means wall, and the extension of the connective tissue can form or contain blood vessels and such. So you can just think of it as an extension of that connective tissue, the capsule going into the tissue itself. I don't know if faculty have anything else that they want to say. And that's common too. If you look at the other histology lessons we have, you'll see kind of patterns like that as well.
Okay, yeah, thank you.
Mhm.
Another question I had is for T lymphocytes, they're like growing in the thymus. Where do they go from there? Do they stay in the thymus, or do they go to like a secondary organ, which is for recognizing the pathogens?
You get those T lymphocytes will circulate. So those would be our T cells, is what we tend to think of those in our white blood cells. So those are kind of circulating constantly throughout the body. But they will be activated under some circumstances when there's an invasion from a pathogen. So they're kind of, yes, they'll circulate to the secondary organs, but that's not where they go to live necessarily. They're all over the place.
Right, and there's within the thymus, you have two different subsets of T lymphocytes. You have what will become the oftentimes referred to as the helper T cells, and you also have the killer T cells. And both of those types of T cells are maturing in the thymus gland. They'll both be released, but there's a pretty stringent, let's call it a screening process in the thymus. And out of all of the lymphocytes that are going there to mature, only about 1% escape out of the thymus and escape the stringent screening process. That's because they recognize the self tissue, and that would lead to autoimmune kind of tissue destruction. So there's a lot of maturation going on in the thymus, but there's also a lot of apoptosis. Once those that have passed that screening process, if you survive, then you'll move into the bloodstream and take up residence in different locations like the spleen, like the lymph nodes, like the tonsils, etc.
Wow, that's awesome. Okay. I think that's most of what I had. Oh, where are the pairs patches? Are they all throughout the gastrointestinal or?
Small intestine. We kind of, I don't know, when I think of like them, I usually think of them along the like the ileum, so the distal small intestine, and I guess we can find them. The appendix is often filled with this kind of tissue as well.
Okay. Oh, sorry. Last question. As far as all these cells that are like in the lymph nodes, are they just kind of floating there, or are they like what are they surrounded by? What do they rest on, and how does all the lymph get filtered through them if they're where they are?
I think it's, let me see if I've got a decent, we kind of want an electron micrograph here, and I'm not sure that I have one for you. Let me see if I can zoom in a little bit on without, yeah, causing too many issues here. There is space between these, aren't tight junctions, so things can move kind of fluidly amongst each other. If we think about the way the, so for say the spleen here, blood can move out and through. It's not this very tight matrix. So it's kind of looser tissue in that sense, but it is relatively densely compacted, just not with tight junctions between each cell.
Yeah, and just really quickly, those trabeculae that you were asking about that Dr. Haggerty explained, they really serve almost like walls or anchors for the core tissue in the cortex. And then there are cells that produce almost like a spider web of fibers. They're called reticulocytes to produce reticular fibers. And that's really what the lymphocytes and the macrophages are anchored onto. So yeah, you're right. It could be like they should just get washed, in the front door and washed right out the back door, but they actually can anchor onto these fibers. It's very similar to like a piece of netting as far as the reticular net-like those fibers within these secondary lymph organs.
That's awesome. Thank you so much.
If you look up like reticulin stains, you'll see there's a decent way of actually visualizing some of those fibers. All right.
Thank you, Gabe, for that presentation. Remember that this is all recorded, so you can go back through this and play it at double and triple time and whatever time you want to listen to it. And you can go back through these explanations. So our last presentation is going to be given by Hannah Lufkin, and she is going to talk to us about the embryology of the head and neck. So Hannah graduated from Michigan State University with a degree in genomics and molecular genetics, and is currently a medical student at the Edward Via College of Osteopathic Medicine in Auburn. In her free time, Hannah spends time with her family, takes her dog Izzy on walks, and listens to Taylor Swift. Hannah's favorite part of anatomy is neuroanatomy. So Hannah, if you would take over and give us the last portion of our presentation this evening.
Sounds good. Then we can go watch Taylor Swift at the Super Bowl. So, you know. Okay, so I'm going to talk to you about head and neck embryology, which can be kind of complicated. So please, let me know if you have any questions and I'll do my best to explain it or pass it off to the doctors who know a lot more than me. So, we're going to start by just kind of taking it pretty simple here. Our learning objectives are defining the pharyngeal arches, the pouches, and the grooves, which sounds very foreign, but we're going to work through it. And you guys will know a lot by the end of this, I hope. So first, we're going to look at the structure, like when do these structures develop? So we're kind of looking at a timeline of the head and neck structures begin developing around week 4 after fertilization, and they continue through embryogenesis through week 8. So there's various steps throughout these 4 weeks where things can develop. So we're going to get looking into those. But first, we can look at like the pharyngeal arches. So here is an embryo. As you guys see my pointer, okay? Yeah. Okay, I just wanted to make sure because I'm going to do a lot of pointing. So here is an embryo. We're looking at it from a lateral perspective, and that's really important here because the pharyngeal arches, which are right here, they arise from the embryo laterally, and they happen since they're lateral, they happen on both sides of your head or on this little embryo on both sides. So they're lateral paired structures. So here we have our gray neural tube, which we know is going to develop into the spinal cord. And then back here we have these R structures, R1 through 7, which are the ribomeres, and they are specialized regions of distal and caudal ends of the developing brainstem. And these ribomeres are really important because that is where the neural crest cells migrate to the pharyngeal arches. And I know that sounds really confusing, but so this is where our neural crest cells begin, and I know you've probably talked about them before, or maybe not, but they're very important for head and neck anatomy, and we'll get into that in a minute. So then our Roman numerals here, 1 through 6, those are our pharyngeal arches. So these are where they would be laterally, and then our neural crest cells, this is a good picture to kind of show it, they migrate from the ribomeres to its perspective, like pharyngeal arch. So the ribomeres of 1 and 2, those neural crest cells will migrate to pharyngeal arch 1. And then the ribomere neural crest cells of 4 migrate to the pharyngeal arch of 2, and then 6 and 7 ribomeres go to pharyngeal arches 3, 4, and 6. You might be wondering where 5 is, and we will get to that. So let's talk about like the parts. So there are 6 pharyngeal arches that develop. So like we said, here's the same embryo. Here's a picture of it from a lateral perspective. There should be 6 of them here. But the 5th one, there's no adult structures that develop, so it kind of just disappears. So it doesn't really make any big kid structures, so it kind of just goes away. And then pharyngeal arch 4 and 5, pharyngeal arch 4 and 6, they kind of combined later throughout development. So even though we have pharyngeal arch 1, 2, 3, 4, and 6, we kind of just say that there's 4, which I checked with Dr. Peterson before this because that still kind of confuses me. And then we also have 4 pouches and 4 clefts or grooves, which I'll talk about on the next slide, but you kind of see them in the picture here. And then, like I said, again, they are bilateral structures, so it happens on both sides of the head and neck. So that's like what they give rise to, like structures in your skull, in your face, and in your neck. So let's talk a little bit about what these fancy words are that I'm saying. So if we look at the big picture of the embryo here, let's pretend that we made a cut where this red line is. So if we're making a cut there, that's what this bigger colorful picture is. And what we're basically looking at is the inside of the embryo if we make that cut there. And in here, we're looking at, this is where the pharynx is, which makes sense because if you're like around the pharynx, that's like head and neck situation. So that kind of helps me remember like where I'm at and like where this is all developing. So the first thing we're going to look at is the pharyngeal pouch, which is kind of this peachy color here. And the pharyngeal pouches are between 2 pharyngeal arches. So So the teal is the arch and the peachy color is our pouch. So the pouches are along the pharynx, so on the inside of the embryo, and they're like the first pouch is located between the first and second arch. So this would be the first pouch between arch 1 and arch 2. So then you can say that there's a second arch, third arch, and fourth arch. So there's or pouch, I'm sorry. Whoops. 1 pouch, 2 pouch, 3, 4. So there's 4 pouches, and those are composed of endoderm. And I like to think of inside endoderm. That just kind of helps me, like on the inside endo. That helps me remember it. So then we have our pharyngeal arches. So that is the teal right here. And those are like kind of swellings, and those are what like arise laterally on both sides of the head and neck. And those are composed of mesoderm and those fancy neural crest cells I was talking about. And then we have our pharyngeal clefts and grooves. It kind of depends on like what source you look at for what word they use. So just be careful with that. And those are, so if we were looking at the external view, lateral perspective of the embryo. So if we're looking at this guy right here, this would be a pharyngeal groove or cleft right here. So this yellow would also be seen right here. So you can see it from the external. Still very small, so it'd be difficult to see without like a microscope, but that's kind of the idea. So then the clefts and grooves arise between an arch. So the first cleft is between the first cleft between the first arch and the second arch. And that is ectoderm, which ecto kind of ectopic outside. That's kind of like how I remember it. It's probably how it was meant to be, but it's nice that it lines up that way because I don't always feel like it does with embryology. So what are some like things that we've talked about, like where they are, but like why does that really matter if we don't know what they become? So we're going to talk a little bit about why these structures are so important and why we care so much about them. So we have our pharyngeal clefts, which as we can remember on the outside, so it's ectoderm, and those make a lot of epithelium. And the first cleft is really responsible for a lot of the skin and lining of the external ear. And then we have our pharyngeal arches. And like we said, this is like those middle swellings, and those are composed of either mesoderm, which is going to make connective tissue, cartilage, and bone of the face and neck. We have muscle of the, or skeletal muscle of the face and the facial muscles, so what allows us to smile. We've got our blood vessels of the head and neck, and some originating from the heart as well. And then we've got our neural crest cells, which are really important for the nerves of the face and the neck. And then lastly, we have our pharyngeal pouches, which is endoderm, so on the inside and like lines the pharynx. And that's responsible for a lot of glands. So then I kind of comprised a little chart of kind of the more important structures. There's a lot of tiny structures sometimes, but we can just kind of walk through this for some important structures. So the first arch is going to do a lot of the bones of the face and jaw. You have the maxillary artery, cranial nerve 5, the second and third branch of that, and then certain muscles. Where cranial nerve 2 is the some ear structures, some facial muscles, and then cranial nerve 3. But remember, all these cranial nerves are going to come from more of the neural crest cells, where the rest of the structures are probably going to arise from the mesoderm because we are in the arches. And then we've got from the third arch, we have part of the hyoid bone, the pharyngeal constrictor muscles, cranial nerve 9, and the common carotid. And then, like I said, 4 and 6 often combine, and they can make really similar structures. So you're going to have cartilage of the thyroid, pharyngeal constrictor muscles, and then our neural crests from 4 are going to give the aortic arch and the superior laryngeal branches of cranial nerve 10. And then 6 is laryngeal or larynx cartilage and muscles, and then the ductus arteriosus and pulmonary trunk. And if we move on to the pouches, so remember that's on the inside, that's from the endoderm and kind of around the pharynx. So you have some middle ear structures, tympanic cavity, and the Eustachian tube. In pouch 2, you're going to have the palatine tonsils and epithelium. And then 3, you're going to have part of the thymus and inferior parathyroid glands. And then 4 and 6, like I said, are going to kind of, there isn't really a 6th one. because they combine together. And they're going to make the parathyroid cells of the thyroid and the superior parathyroid glands. And then for the clefts, let's remember that's the external, so we can see it from the outside. So that's going to be ectoderm. And this, like I said in the previous slide, the first cleft is going to give rise to external ear structures. 2, 3, and 4 are all going to give rise to cervical sinuses, but these obliterate during week 7, so they don't really have any permanent structures. And that is all. I know it was a lot. So let me know if you have any questions. Hopefully I can answer them.
Okay, Michael, you can go ahead.
Okay, first question, actually, I know it dissolves before adulthood, but what was the point of the fifth number 5 that disappeared? I think it's arch.
Yeah. I guess I'm not really sure where. I think it just kind of dissolves and then 4 and 6 combined, but I don't have a great answer for that. I don't know if Dr. Peterson has a better answer than that.
It's really kind of minuscule, and it mostly, it just gets overgrown by cells that are rapidly dividing in the other areas of this. This whole thing is called the pharyngeal apparatus. So in other, vertebrates, it has a more significant role, but certainly not in primates or humans. And so I'm seeing William's question in the chat. So, and you're making me laugh, William. Exactly. His question was, do we really need to know all that stuff that was on the chart? And the answer is, Hannah, put that in there so that you have something to kind of have a reference for, but no, you need a basic understanding of the difference between a pouch, an arch, and a cleft, and kind of have a sense of which of those that we just talked about are comprised of endoderm versus mesoderm and neural crest cells versus ectoderm. If you get that much, I would be so happy.
And Michael, did you have another question?
Yeah, I'll ask another one. What is embryogenesis? Because I know it happens in week 8. I think it said on the first slide, but I was just kind of curious. That does make me feel a lot better that we don't have to learn all of this stuff.
Yeah, I just kind of put that in there because to me, like, I don't know, people might be curious about what these big things rise to. So sometimes that helps, but yeah, they're a lot to take in. Embryogenesis, like, I think basically as your embryogenesis is like fertilization to week 8 is my understanding, is like just how the embryo arrives. And then I think you go into the fetal period, correct? Is then the like around week 8, it's fetus until birth. And so it's just the period of when you're an embryo to a fetus. Is that weird structure the eye? No, it's not the eye. Isn't it the otic cavity? Isn't this like the otic cavity or something?
The otic pit.
Yes.
I think that is becoming from the diencephalon region. So prosencephalon, Hannah, you know all this. Prosencephalon, and we're going into telencephalon and diencephalon, and that would be the optic, right? The optic disc forming, optic vesicle. The otic should be over here. Kind of, I know you can't see my cursor, but that's the other weird little thing that looks like an eye, kind of way over there laterally. And then as the head grows, the ear kind of comes up from low in the neck to sitting on the head.
Okay, is there any other questions? All right, I want to thank you all for coming to tonight's session. I just admit somebody in. Just remember, this is all being recorded. It will be posted shortly, in the next at least 24 to 48 hours. So feel free to go to our YouTube channel and check out the recording itself. As a reminder, next week is the last review session before the local competition. And so we will have our last session on Sunday, February 18th, starting at 6 p.m. It'll be held by yours truly, Drs. Haggerty, Peterson, and Piscura. So we can have a nice Q&A before the local competition. And then the other thing is that tomorrow, Monday, February 12th, will be the last day to register for the 2024 competition. So if you have not registered yet, make sure you do so. And with that, I thank all the presenters and everybody coming tonight. Have a good night.
Good luck, everyone.
Good luck.